This is the second part of our second lecture set on what we're calling allelic interactions. And we're labeling more allelic interactions because we've been talking about interactions for a couple of lectures. The last one we talked about lethality. We talked about the concept of a dominant lethal and a recessive lethal. And we talked about a new term called pleiotropy. And pleiotropy or pleiotropy is where you have a single gene because it impacts a number of pathways affecting one or more traits. And in fact, pleiotropy is more than one trait. So we showed a chart something like this. We explained that lethal genes were those that caused death. We talked about what a dominant lethal was and the fact that either in a homozygous or heterozygous condition, it would be a lethal, and why some of those might persist. And then I want to talk a little bit about a recessive lethal, which are actually the more common types of lethals that we might see. So we'll just use kind of a strange little example here. Back in the 1920s, fox furs were very, very common, very important, and there was a lot, had been for many centuries actually, fox collections. People then had started to, by this time, had started to actually rear foxes in captivity and enclosures for their furs. And so they were very valuable, and they make very beautiful coats that you kind of see. And of course, you can even get silly things like hats. But there are a couple of different colors that are very popular, and this kind of illustrates one example right here. One of them is called silver, which is this example that we see here. And then there's another color called platinum, which are these beautiful white ones. The platinum ones were actually originally not very popular with the people that were doing the breeding because they were difficult to maintain and had some issues, and we'll see why in a moment. The silvers are, but the platinums are very valuable types of furs. Of course, foxes in general are these sort of natural red colors that they have. So let's kind of get away from the silliness and talk about what lies underneath this. Well, it turns out that there's a very straightforward recessive lethal condition found in these coloration in these foxes. Okay, um, and if you were sitting there doing breeding experiments, if you took a silver times a silver, you would always get silver. Okay, so it appears that silver could breed true, right? If you took a silver times a platinum and made that cross, okay, so you have this little silver and this light colored platinum, you always ended up with a 1-to-1 ratio of silver to platinum. Okay, all right. And then if you cross the platinum with the platinum, we see a new ratio for us called a 2-to-1, right? A 2-to-1 ratio is not something we've seen so far. We've seen a 1-to-2-to-1, and we've certainly seen 1-to-1 and 3-to-1s and these things, but not a phenotypic outcome ratio of 2-to-1. So let's get away from these sort of pictures and think about really what's the genetics underneath this. Well, if silver times silver always produces silver, or anything that's always crossed with the same form, wrinkled times wrinkled always give you wrinkled, you would think that both of these are homozygous, right? They're true breeders. And that's exactly what we have here. We have a silver allele, and we have a homozygous condition in this first part, always going to produce silver, right? They're all silver. So that's just true breeding, okay? Silver then is in this little s condition over here, okay? And we're trying to figure out how we can get this ratio of a 1-to-1 of silver to platinum, right? Well, silver is this homozygous condition, okay? We'll just call it S's. You call it little A's if you want or whatever. But notice we don't get 3-to-1s, we don't get 1-to-2-to-1s, we get something else. The reason for that is under recessive lethals, this platinum right here that we're putting in the second one is always a heterozygote. Now you can think of it as big A, little a, or whatever you want, big S, little s, doesn't matter. I'm just going to put the big P, and I'm going to make it red to indicate that there's something else going on with it, okay? That something else is going on is pleiotropy, right? The color here is involved. But if part of that production of that color, we end up with this sort of lethal condition. Now it's a recessive lethal, not, okay, if it were a dominant lethal, these platinums would die, right? Either at some point at birth or later on, but it's a recessive lethal. So they're completely normal individuals. There's just like any other recessive, okay? It's going on and producing its normal function, okay? Platinums, if you notice here, we get a 2-to-1 ratio, right? So what we always get when we cross that which means if you look back at Mendel's Big Six, right? If we go back to Mendel's Big Six, and this is nothing, even though it's lethals, it's not anything different. What produces a 2 to 1, the only thing in here is actually a 1 to 2 to 1 produced out of a heterozygous cross, right? So what that infers and what's actually going on here is that all platinums are heterozygous. Okay, so if you cross a het times a het, we know that we're going to get a 1 to 2 to 1 ratio. It's just Mendel's Big Six, right? Except in this case, one of those is going to die out on us, right? We're going to lose them. So by the time we get to actually observing the phenotype, so we can tell, okay, who's platinum and who's silver after birth, we really only have this 2 to 1 ratio, right? So the 2 to 1 becomes a critical ratio for us to get. So once again, this is a example of pleiotropy. Color here is part of this whole system, right? And we have color and then life, right? So we have the extreme cases of pleiotropy. Now, we're introducing a very important ratio. You see 2 to 1, you can figure that you're dealing with a recessive lethal, okay? If you see a 2 to 1 ratio, you've got a recessive lethal going on. And so the 2 will be associated with whatever is a heterozygote, and the 1 will be one of the other homozygotes. Okay, it won't really matter too much unless there's other information, but you'll know that it's part, in this case, part of the little s, the silvers, but it could have been the big P, whatever. But it's homozygote related to 1. Yes, so we're making a 1 to 2 to 1, but we're just getting rid of one of the forms. There are a number of different types of recessive lethals that are out there in the laboratory mouse, which is originally from a house mouse. There's a condition known as a Goody, which is the common gray form that you probably see maybe running around in your neighborhood every now and then of these musks. There's a yellow, which is a recessive lethal, and the yellows are only heterozygotes. In cattle, there's something known as dwarf compost, and dwarf compost is a recessive lethal, and there's only heterozygotes there. Hairless, like chihuahuas and things, many of those forms are really a recessive lethal, so all the hairless individuals are heterozygotes, and if you cross them, two of the hairless, you often end up with a lot of stillborns and other things in the litter. That's because part of them are these homozygous recessives that you're producing. And then in horses, there's variation. Again, roan, we keep going back to that as a variation, but there's some roan that's in fact a recessive lethal. And you cross two roans, you expect to get a 1 to 2 to 1, but you end up with a 2 to 1 ratio because there's lots of lethality there. Okay. One other example that kind of pulls all this together is a Minx Cat. A Minx Cat is the one with the tailless version of cats, and there's classic pleiotropy here. The tail condition is a dominant allele, right? It's basically there will form, but it at the same time is a recessive lethal in these particular breed of cats. Okay, it has, just to remind you of some terms, it has high penetrance. When we talk about lethals, we start really talking about penetrance, right? Penetrance is if you get the allele or you get the genotype, are you actually going to express it? And this one has very close to 100% penetrance. So basically, if you get the allele, the combination, you're going to see it. And what you see is a cross that looks something like this, okay? A Minx Cat has to be a heterozygote, so your heterozygotes for normal or absence of a tail, normal, absence of a tail, so het times het. You do a little Punnett square with that, and what you end up with is this A allele is a lethal, so you lose those. You get two Minx Cats out of it, and these are heterozygotes. And then finally, you get a normal cat that's a homozygote for the normal allele. So again, if you look at this Minx Cat, you know it's a heterozygote. You know immediately what its genotype is relative to this particular allelic system. Right? Well, that's a quick run through of what recessive lethals are. Fairly straightforward. The basic idea is it takes 2 copies of them to kill you. And it doesn't matter what that allele is doing. It could be doing something codominantly or dominantly or incomplete, whatever. Once you get 2 of them together, though, of that particular mutation of that allelic form, you end up having a lethal condition. Okay? So now that we've got dominant and lethal going on here, it's probably good to go and try a couple of problems working with these and see how we can put those into interplay. So in part 3, as we often do, I'm going to work through a series of problem sets that involve lethals.